Hi, good morning students. And Are you all fine in this lockdown and enjoying very well at home while studying and watching my videos on YouTube? And this is my channel. Again, I am going to introduce the channel name is Ostia Academy. Okay, this is my channel and this is in front of you your biology teacher, Mr. Kumar Animesh. In previous sections, in previous videos, you have learnt about genetic chapter and the chapter name is Principle of Inheritance. Okay, there are different types of inheritance has been explained by me in different videos. Now today I am going to tell you about the next topic of this chapter, that is qualitative inheritance. Okay, qualitative inheritance. What does it mean? Qualitative inheritance and quantitative inheritance. There are two different types of inheritances, namely qualitative and quantitative inheritance. Okay, so now let's start to the topic, that is qualitative inheritance. Firstly, I would like to explain the meaning of this inheritance, qualitative inheritance. What does it mean? It is the type of inheritance in which a single dominant gene influences a complete trait. Presence of two such dominant genes does not alter the phenotype. Okay? With a suitable example, the genes controlling inheritance are called monogenes. Examples are capital T or capital T small t for tallness in pea plant. Okay, so these are the example of monogenes. Here is clear, you can understand very well with the help of dominant character, that is the tallness of pea plant, which is represented by capital T alleles. Okay, and capital T small t alleles. These both are alleles for tallness in pea plant. Okay, the first one is both capital represent the homozygous and capital T and one small t together represent the heterozygous. Okay, it means that qualitative inheritance, in other hand, you can understand well, in qualitative inheritance, it is the type of inheritance in which a single dominant, only one dominant gene influences a complete trait. Only one, that is capital T, which is responsible for tallness of plant, the only single gene. Okay. Now, the second point is that qualitative inheritance produces a sort of discontinuous trait variations. Okay, qualitative inheritance produces a sort of discontinuous trait variations in progeny. Example, either tallness or dwarfness. Clear? Discontinuous, not continuous, tallness. These are the perpetuation of tallness haven't been found in this type of qualitative inheritance. Okay, it leads the alternative or discontinuous trait of variations, that like in F1 generation tallness, okay, in F2 generation both tallness and dwarfness have been found after the cross between F1 generation. Okay, now intermediate forms or continuous trait variations are not produced, in which that is qualitative inheritance. Clear? Also called monogenic inheritance. So these are the some points which deals the qualitative inheritance with suitable example. Understood? I think you well understood. Now we will move on second topic, that is quantitative inheritance. Quantitative. It was first studied by J. Col Reuter 1760. In case of tobacco and F. Galtron 1883 in case of human beings. Okay, both the scientists together had studied about quantitative inheritance in tobacco and human beings. Clear? Nielsen Hell obtained the first experimental proof of polygenic inheritance in case of kernel color in wheat. Okay, kernel color in wheat. Wheat is a crop. It means it is a type of inheritance. Now we are discussing it is a type of inheritance controlled by one or more genes in which the dominant alleles have cumulative effect, which each dominant allele expressing a part or unit of the trait. The full trait being shown only when all the dominant alleles are present. Okay, so here you can say this is a type of inheritance in which one or more genes in which the dominant alleles have cumulative effect, which each dominant alleles expressing a part or unit of the trait. Clear? When the trait being shown only when all the dominant alleles are present. The genes involved in quantitative inheritance called polygenes. It means there are two different types of genes. That's why the term it is also called as polygenes. This is also called polygenic inheritance. Clear? The traits controlled by quantitative inheritance are sometimes known as the trait controlled by quantitative inheritance are sometimes known as eccentric traits because they can be measured in terms of unit of size, height, weight or number. Now here is some examples: kernel color in wheat, lobe length in maize, skin color in human beings, human intelligence, milk and meat yield in animals, height in human beings, and several plants. These are some examples of quantitative inheritance. Here is a graph, histograms showing the distribution of apto phenotype in case of polygenic inheritance. One polygene pair, two polygene pair and three polygene pair. Here is a histogram, and with the help of this histogram you can understand very well of quantitative inheritance. Now polygenic inheritance can be known from the frequency distribution of phenotypes. In monogenic or qualitative inheritance, the phenotypes are 3:1 in case of single gene pair and 9:3:3:1 in case of two pairs of genes. That is the Mendel's law. Already you have learnt in previous sections. That is the monogenic in case of monogenic or qualitative inheritance. In polygenic inheritance, the number of phenotypes is 3, 1:3:1 in case of one gene pair, and 5, 1:4:6:4:1 in case of two polygene pairs, and 7, 1:6:15:20 15:6:1 when three polygene pairs are involved. So these are some examples of polygenic and monogenic inheritance. Thus we see that the number of inheritance and intermediate types increases with the increase in the number of polygenes, but the number of parental types remain the same. Now you can understand with the help of histogram. The possible origin of polygenes is duplication of chromosomes part, polyploidy, mutations producing genes having similar effect. Okay, and these are the some difference between monogenetic qualitative and polygenic quantitative inheritance as representing found respectively found in pea by Mendel and tobacco by Carl Reuter. These are the some examples. Okay, now here is a question, and the differences between monogenes and polygenes. You have to learn this. Okay, the differences between monogenes and polygenes. The first I have bifurcated in two halves of the first one is monogenes and second hand in polygenes. Okay, monogenes is also known as monogenic inheritance and qualitative inheritance, and polygenes, these are the quantitative inheritance and polygenic inheritance. Now we can study one by one. They produce discontinuous variation in the expression of traits. Which one? Monogenes. They produce continuous variation in the expression of traits. A single dominant allele expresses the complete trait. A single dominant allele expresses only a unit of the trait. Monogenic inheritance controls qualitative traits. Polygenic inheritance controls quantitative or metric trait. Okay, a character is represented in an individual by a pair of alleles. A character is represented by several pairs of alleles. F1 individual to similar to dominant parent. F1 individuals are intermediate between the two parents. Okay, so F2 individuals resemble both the parents in the ratio of 3:1 depending upon the number of polygenes. Okay, number of polygenes, 2/4 on pair, 2/16 2 pairs, or 2/64 3 pairs. F2 individuals resembles. No intermediate are produced, the parental type. Intermediates are quite common in polygenic inheritance. Okay, the number commutative action in the presence of two dominant genes. The dominant genes have cumulative effect on the expression of the trait. Individuals with dominant phenotype are more numerous than with representation of phenotypes character. Individuals with dominant trait are usually as few as with a recessive trait. Okay, and in this the individuals are with the recessive phenotypes. Okay, so these are the differences between monogenic and polygenic inheritance. Okay, now here is some example of human skin color. What are these? Now we can study human skin color. The next topic is human skin color. It was first studied by Davenport 1930. In case of Negro Caucasian intermarriage in Janica and Buddha. Human skin color is caused by pigment called melanin. Okay, I think you well familiar with the term melanin. Melanin is a pigment which is found in the skin of human being. Human skin color is caused by pigment called melanin. The quantity of melanin is due to 3 pairs of polygenes. Okay, 3 pairs of polygenes, A, B and C, 3 pairs. That's why this is polygenic inheritance. If black or very dark, then the alleles are capital A, capital A, capital B, capital B, capital C, capital C. And white or very light, all are in small alphabets. small a, small a, small b, and small c. Individuals marry. The offsprings or individuals of F1 generation shows intermediate color, often called mulatto. Okay, often called mulatto, and the genotypes are capital A, small a, capital B, small b, capital C, small c. These are the alleles. When two such individuals of intermediate color marry, the skin color of the children will vary from very dark or black to very light or white. A total of 8 allele combination is possible in the gametes, forming 27 distinct genotypes distributed into 7 phenotypes. Okay. A histogram prepared from the frequencies of various phenotypes shows a bell-shaped curve. That's why the histogram that I represented in next page. From this histogram you can understand very well. Now here is a cross which is given in this video. Here you can see very easy and understand very well with the help of this representation, and this representation is based on quantitative inheritance of skin color in human beings. The first one, the very light, the one parent is very light and second parent is very dark. The offspring will be mulatto. Okay, mulatto. Gamete, one gamete, one gamete. You can understand. Already you have understood very well the dominance law of inheritance and Mendelism. Okay, with the help of the previous knowledge you can represent the different flow chart and understand very well with after seeing this video. Okay, so these are the representation of human skin color cross. Okay, in next, histogram and bell-shaped curve produced from the frequency of various skin phenotypes produced in F2 generation after a cross between black and white individuals. Okay, sometimes in NEET examination the question has been asked on the basis of this histogram. Okay, sometimes already in this question has been asked what is this histogram shape of quantitative inheritance. Okay, so this question has been asked in NEET examination. So these are the inheritance that you have learned and you have watched in this video. Now we are going to discuss next topic, that is pedigree analysis. Pedigree analysis. What is the meaning of pedigree analysis? This is a family tree of human being, a record of inheritance of certain genetic traits for 2 or more generations, presented in the form of a diagram or family tree, is called pedigree. Clear? It is a inheritance of certain genetic traits. Okay, from 1, 2, 3 generations, which are represented the 1 to more than 2 generations. This is known as pedigree or family tree. It is employed in case of human beings and domesticated animals, especially pets. Some symbols are given here: male for square, female for circle, carrier male for solid square, and carrier for solid circle, and first and second offspring with Arabic numerals, first and second. These are the in numeric form, Arabic numerals. Pedigree analysis is a study of pedigree for the transmission of particular trait and finding the possibility of absence or presence of that trait in homozygous or heterozygous state in a particular individuals. It is useful for the genetic counselors to advise intending couples about the possibility of having children with genetic defects like hemophilia, color blindness, alkaptonuria, phenylketonuria, thalassemia, sickle cell anemia, paracytactyly, and syndactyly. Okay, dominant. These are dominant, and sickle cell anemia is a recessive form. Pedigree analysis employs two tools: principle of probability and chances of differences in realized ratio due to smallness of the progeny. Okay. Second one, elimination of alternations and alternatives. A few problems of autosomal inheritance are given, and that I have given in my notes that you will understand. Okay. First one is a problem 1. In the pedigree given below indicates whether the shaded symbols belong to dominant or recessive alleles. Also give genotype of the whole pedigree. Now, with the help of this diagram, you can understand the first one is this one is a square for male, and this shaded square is a carrier. Okay, and with which married with a normal female. Now, in F1 generation, this is a Arabic numeral which shows the generation, and in F1 generation, they have one female carrier, one male normal, and one female normal, and one male carrier. After marriage between one carrier with a normal male, that after that they produce one carrier female and one normal female in second generation. Okay. Now, with the help of this diagram, you can understand well. Since the shaded symbol appears in all the offspring, father must be homozygous dominant, while the mother homozygous recessive. Okay, all A. Because in all other cases, this possibility is absent. Okay, here is given. All the number of second generations will therefore be heterozygous. This is further confirmed by marriage of second one with homozygous recessive, okay, and being children of both the parental types. Marriage of third with the homozygous recessive can be produced both recessive and heterozygous, or as present here. Okay, so these are the examples and these are the problems which is based on the pedigree analysis example. Okay, so this is the last topic of this chapter, and I think you have understood very well, and keep watching my video. Bye bye. Thank you.